My name is Peter Bell. I'm Executive Vice President at Autism Speaks for Programs and Services. I'm also the chair of the Community Advocacy Committee, which is part of IMFAR, which is helping to sponsor this luncheon. This is the second year that we've done this. Last year we had about 60 people, and this year, based on the number of 90 lunches that are still there, we probably have about 80 people this year. So it sounds like we're very consistent with the growth of IMFAR every year. We decided to do this luncheon because we knew that a number of people come to IMFAR every year who have family members that are on the spectrum. They may be individuals themselves that have autism, or they may be just someone who cares very deeply about someone with autism. And we wanted to make IMFAR a welcoming place for those individuals. Some of them may be scientists, some of them may be just people who are interested in learning about the science. Whatever the reason might be, we are really thrilled that you're here today to learn more about some of the great science that's being presented this week here at the 10th Annual IMFAR. Last year, I think we had enough people where we could kind of go around the room real quickly. I don't think we're going to be able to do that, but what I'd like to do is just a show of hands. How many of you are parents of a child with autism? So probably about 3/4. How many of you are an individual that has autism? So about 10 or so. That's great. How many of you have a sibling that has autism? Nice. That's great. And how many of you are just someone who wants to know more and are here as a professional or good, excellent, wonderful. So, and how many of you are here just for the free lunch?
Is there extra seating here?
They're here. You can sit here.
Okay.
Just a couple things before I introduce our panel of scientists. I think in most of your tables you have these purple ribbons. We decided that we wanted to have a way in which we can identify one another. It says stakeholder on here. So if you don't have one on your name tag already, feel free to, you just pull off the sticky thing and you stick it underneath. There are also some puzzle piece pins that were actually made by a family that has children with autism. They generously donated those, and it's another way to identify yourself as a stakeholder within the community. So without any further ado, we're going to divide this up into two different panels, so to speak. The first one will be three scientists who are here either very much involved with the meeting, making presentations and so forth, or are very active within our community. And then we'll open it up after their three presentations, do a little Q&A, and then we'll finish off with a second panel where we have both a parent who has a child with autism as well as an individual with autism who will share some of their perspectives on what the meeting has meant to them so far. So I'm really honored to be able to introduce all of our speakers, but certainly David Amaral, who will be our first speaker, is the outgoing president of IMFAR, the Society for Autism Research. And David is also a professor, a distinguished professor, I want to make sure I get it all correct, in the Department of Psychiatry and Behavioral Services, and also the Director of Research for the UC Davis Mind Institute. David, along with a number of his colleagues, were very instrumental in founding the Mind Institute, probably, what's it been, about 12 years, 10 or 12 years? 12 years, 12 years. And he also was, although he's very modest about this, he was very instrumental in the starting of IMFAR itself. We honored the founders of Cure Autism Now and the National Alliance for Autism Research yesterday with the IMFAR Advocate Award, but David was very much involved with both Eric London and Portia Iverson in helping to start this meeting 10 years ago. So we're very lucky to have David here with us today. I think he's going to share some perspectives of what he sees, some of the good science that's been presented this week, and maybe perhaps where we're going. I don't know. We hadn't really planned anything specific, so why don't you come on up, David, and share some thoughts with us.
I thought I would just start off by saying a little bit about this meeting. And since my presidency is coming to an end, I've been sort of thinking about it. And I think if my board of directors has tried to do anything, is we've tried to be as inclusive as possible for this meeting and to actually reduce stress and reduce tension. And so I hope that this meeting, this luncheon, actually is doing a little bit of that for you. This is a scientific meeting. We started 10 years ago, as you know. We thought it was a huge success when we had, I think, something like 220 people come the first year. And now it'll be something on the order of, I think we're at around 1,900 people attending this session, this meeting. So it's grown enormously. And it really is first and foremost a scientific meeting. And so there's been a tension over the years of saying, well, should we let any stakeholders come to the meeting at all? And I'm sure you've all heard this before. Scientists like to communicate at sort of a high level in a lot of jargon, and often don't want to present their science to a lay audience at these kinds of meetings. And so we've sort of had to honor the peccadillos of scientists and make sure that we continue to have this meeting be pretty much focused on the highest level of science. But at the same time, most of us, as I said at the awards ceremony yesterday, most of us wouldn't be here if it weren't for parent advocates and for the stakeholders in the autism community. So I think we have to do whatever we can to acknowledge that contribution that the stakeholders make and try and translate information to the broader community. So we've done it in this way by having Peter develop this community advisory committee, and I think this committee will continue to develop ways to try and convey information. We've done it by developing, Allison Singer and Dana Marnane have a public relations committee so that we can actually try and translate some of the hottest findings out to the community. So we're trying, and again, I think we want to be as inclusive as possible. So what's changed over the 10 years? Well, I think we have, again, been much more inclusive. When the meeting first started, there were a lot of schisms. I won't go into them, just, it's like salt and wounds, but there were a lot of schisms. And what I've seen over 10 years is those schisms have largely healed. Not totally, but largely healed. So we have advocacy groups coming together. We have groups who thought, it's all the environment, or groups that think it's all genetics, sort of come together and now thinking, well, maybe it's genes and environment. And it's my hope that this meeting really will embody all the thoughts about autism, all the thoughts about doing research in autism, all the thoughts about doing treatment of autism that are out there. I think the bottom line, my take on the world, is that we still have such a low level of understanding of this disorder that, if somebody has what seems to be a far-fetched or sort of outlier thought about autism, maybe it's going to be true. We thought 5 years ago that you could never treat a mature brain that had a neurodevelopmental disorder. But now we know that there are mouse models that can be completely recuperated based on interventions. So I think in science, you have to be very open-minded, particularly when we have as little knowledge as we have. The other tension that I detect that I hope we're going to be able to figure out, and I was talking to Virginia Bovell just before we got started, and I think that this is a real tension, and you folks have to help us scientists figure this out. And that is to understand the biology, to understand what causes autism spectrum disorders. It's actually easiest to study individuals at the earliest phase of life. I don't know, did any of you see Eric Courchain's presentation? Okay, well, Eric, that's a good example of where he's thinking things are happening very, very early, which then go on to lead to autism spectrum disorders. If you looked at his charts, what you see is that brain development in autism changes. So there's overgrowth early on, and then there's actually a diminishing of the brain size as the person with autism gets older. We want to know what's happening at the very beginning of autism. So there's been a pressure on the field to study infants and even younger and younger infants. So where's the tension? Well, the tension is that now we've had 20 years of an increased number of individuals with autism, and they're becoming adolescents and they're becoming young adults. And people are saying, you know, we need to do more for these individuals. We need to do better treatment. We have to get better residential facilities. And I was at a meeting at the White House just 3 weeks ago, and we were having a nice luncheon, and some of the adults on the spectrum were saying, you know, we have to shift the research agenda away from kids with autism to adults with autism. And I thought, what a disaster. If we shift, we're going to be losing the possibility of getting to the roots of autism. We're only going to get to the roots of autism by studying even younger and younger kids. So what I said was, let's not shift, let's expand and encompass adults with autism, right? So I think as you lobby, and I know the autism community is very good at lobbying, think about the fact that I think the goal is that we want to decrease disability in individuals with autism. We want to get the best quality of life for individuals with autism. And in part, that's going to be by detecting it early on and by either doing preventative measures or interventions. And if we shift our perspective to those that have autism, my view is that, you know, the tsunami of individuals with autism is just going to continue to get larger and larger. So where we need the lobbying is not for a shift, but for an expansion. And at these times of, you know, decreasing economic or difficult economic times, it's going to be hard to do. But I would ask you to consider, as you do your advocacy, the implications of shifting away from basic science into therapy, or shifting into the adults from young kids, because I think that's probably not going to be the right direction to go. And again, what we've seen in this meeting, and this will be my last comment, is that that autism is a lifespan disorder. And it is true that we don't know very much about adults with autism or adolescents with autism. We need to do more research there. I think it's incredibly important. And it's, again, I would be the last to say that if we know something about kids with autism, that's going to give us knowledge about somebody who's 40 with autism. But knowing that it's a lifespan disorder, we have to, again, dedicate more time, effort, and money to studying the entire lifespan as opposed to switching away from the youngest kids. So that's my take-home message. I think, you know, by and large, this meeting has had the best vibe of any of the meetings that I've come to. And the other thing I should say is that more than half of the members of IMFAR and who come to these meetings are students. They're graduate students and they're postdocs. So I think that what that says is that we have a very bright future in autism research. Some of the best and brightest. The next guy that you're going to be hearing from, Ricardo Dolmich, genius. I mean, it doesn't just superior scientist. And so, yeah, you better say something really, really good. Anyway, thanks, Peter, and yeah.
John Elder Robison, who's the co-chair, Sarah Logan over here, who's going to be speaking a little bit later, and Virginia Bovel over here, our international representative from the UK. And then we have 3 other members, Stephen Shore, who was here yesterday and had to take a red eye home last night. He's another, like John, an individual with autism. And also Mark Shen, who I don't think I see, but he may have double dipped on lunches today, and he may be coming over from the diversity luncheon. So I wanted to say thank you to my colleagues on the committee who've been wonderfully supportive over the past year. Okay, so we're going to go to our second, and David gave you a little bit of a precursor to our next presenter. Dr. Ricardo Dolmich is one of the keynote speakers here this week. He'll be making his presentation tomorrow on pluripotent stem cells, which is an exciting area of development. I'm sure he's going to share a little bit about that. David did say that this guy is a very bright guy. He is officially the associate professor in the Department of Neurobiology at Stanford University. He has places like Brown and Harvard in his CV as well. He has received a number of awards and honors during his very short, distinguished career, including the Society for Neuroscience Young Investigator Award, which is a really big deal, an NIH Pioneer Award. He's a Searle Scholar. He's a McKnight Scholar. And he also received a career award from the Burroughs Wellcome Fund for Biomedical Sciences. And I guess what's also unique about Ricardo is that he's also a stakeholder. He has a son who I think has been his inspiration that has autism, and hopefully he'll share a little bit about that with us as well. So without any further ado, Dr. Ricardo Dolmich.
Okay, so I think what you're going to see in a second is that I'm a very good argument for this idea that autism is genetic. I also don't know if I can live up to that introduction. So I guess I should start by telling you why I became interested in autism. I actually started my academic career far away from this. I basically studied biophysics. And I was interested in interesting and what I thought were very important but very esoteric questions about how proteins and membranes conveyed signals into neurons, and I was very happy doing that. But you know, about maybe now 6 or 7 years ago, my oldest son was diagnosed with autism, and you know, at first, I didn't exactly know what to do about it. I think our reaction, my wife and I, was probably similar to many of your reactions. I sort of felt, well, you know, we can't leave any stone unturned. We don't really know what this is. So we went off to learn as much as we could, and we tried all sorts of things. And, you know, diets and interventions. And you know, to be honest, none of them worked all that well. I, of course, am a scientist, so I did what I normally do when I'm really stressed, which is I start reading all sorts of stuff, some of it important and some of it not important, and I sort of started reading some of the autism literature. And you know, I realized that a lot of people study autism, but at that time, really very little, I mean, it's still true, but it was much truer then, very little was known about the underlying biological basis of autism. So together with my wife, who's also a scientist, we sat down to think about, you know, whether there was something else we could do. And so sort of at around that time, there was kind of a revolution in genetics. You know, so we have known for more than 30 years that there is sort of genetic susceptibility to autism. We know this from twin studies. So in the 1970s, Louise Rutter and her colleagues published a series of seminal papers which sort of showed that if you compare identical twins to non-identical twins with autism, the concordance in identical twins is really high. In those papers, it was around 90%. Now it's maybe 60 or 70%, but it's still very high. And if you look at discordant twins, that is to say twins that don't share all of their genetic material, the concordance was only around 6%. So this sort of suggested that there was a genetic basis to it. But in fact, we were not good at identifying genes for a really long time. And it wasn't really until sort of what I described as kind of the genetic revolution, which sort of started with the sequencing of the human genome, but really picked up speed with the development of chips that let you look at the whole genome at once, that genes were first identified. And now the pace of identification of genetic susceptibility genes is amazing. I mean, now we can sequence whole genomes for, you know, $2,000. I should just say that the first genome cost $2 billion. So it's great. We're identifying genes at this prodigious rate. Now, the question is, how do you convert a whole bunch of susceptibility genes to a treatment? And you know, I really cared about this, and I thought about it for a long time. And you know, knowing the mutations is kind of like knowing the broken parts list, but not having access to the car. You know, so you know what's broken, but you don't quite understand how the car works, and that's all you have. And so we thought for a while about how is it that you actually go from a mutated gene to a complex behavioral disorder like autism. And so, you know, sort of at the simplest level, right, what happens is that, you know, you have a mutated gene, and that gene gives rise to a protein that doesn't work right, and that in turn changes the function of a cell or a set of cells, and that changes circuits in the brain that ultimately change behavior. Okay, so the standard model for how you go from a gene to function has been to make animal models. Okay, and we've been very successful at doing this for what are called monogenic diseases. That is to say, diseases caused by single mutations. And we can do this because we have all this technology that allows us to change the genetic material in a mouse or in a fly, and we can study those. And we have made mice. And they're very valuable for a lot of things. But it turns out that autism isn't quite like that. You know, it is not a disease caused by a single gene, right? It is this combination of genes. You know, I talk about susceptibility genes because that's what they are. None of them actually give you the disease. They just increase the chances that you might get it. And so one of the reasons for this is that you need to have a bunch of mutations. And so you can make a mouse with one mutation. You can make a mouse maybe with 2 or 3, but you can't make a mouse with 10. And furthermore, we know that the background is important. And as you can see from the fact that I'm here and I kind of move a lot and I have funny movements, you know, background matters. You know, so we needed to be able to do this in people, right? Now the problem is, and that's sort of reason number one. Now reason number two is that, in fact, we haven't been very good at translating things that we have discovered in animals into humans. I have a colleague at Stanford who studies multiple sclerosis, and he tells me, well, you know, if you're going to get multiple sclerosis, the thing to do is to be a mouse. And so it's true, you know, because we've been able to cure mice with multiple sclerosis really well. But we haven't been able to translate that super well into people. So we need to find some way of being able to study the developing brain of somebody with autism. Now, there are sort of two approaches to this. One approach is you can use imaging, and imaging has given us lots of insights. But the thing is that to develop a drug, you need to have access to the biochemistry, to the cells. You need to have something you can screen, right? You need to have some assay that you can test thousands of drugs, millions of drugs, right? And you can modify them, and then you can try them again if you want to develop a drug, or if you, or if you want to use a growth factor, you need to be able to test a lot of them. And so we need something that is easily manipulated, and you can't screen a mouse. You can't screen a monkey. You have to screen a cell or a molecule. So this was a problem because we don't have access to the tissue that is involved, right? We don't have access to the brain. You can't go into the brain of some child with autism and, you know, drill a hole and get some neurons out and figure out why they're broken. And this seemed to be the end of that idea until there was kind of a remarkable development in stem cell research. And this was about 5 years ago. And so, you know, we all come from stem cells, right? So stem cells are these cells that sort of by definition can form every other cell in your body. They're called pluripotent stem cells because they can make everything else. And they're formed very early after an egg and a sperm come together. And we used to think that this was sort of a one-directional process. You start with stem cells and you end up with a human, and it's like spilling water. You can spill water, but you don't unspill water, right? But it turns out that that's sort of remarkably not true. You can take a cell, a differentiated cell like a skin cell, and a skin cell is really, really specialized to be a skin cell. It is not an eye cell, it is not a liver cell, it is not a nose cell, it is a skin cell, right? And you can put into that cell these genes that reverse the process so that now that cell starts morphing and changing its DNA and it becomes a stem cell. Now the thing that makes this powerful is that the DNA in your brain is exactly the same DNA that you have in your cells, right, in your skin cells. So in principle, you can now take a cell that we have access to, like for example skin or blood, and you can take that cell and you can make it into a stem cell. Now, why would that be useful? Well, the other thing that we can do, and here we're kind of sitting on the shoulders of giants, is we can coax these cells into making little brains in the lab. And that seems kind of amazing, and frankly it is. And part of this is that people have been studying mouse development for a really long time. And so we know a lot about the growth factors that are important for forming early bits of the nervous system. And also, just once you put a cell in a particular direction, it tends to form all the other cells that are important for forming a tissue. And so you can make, in principle, you can take these stem cells and make little brains out of them. And you can also make little hearts. And so we decided, I mean, when this was published, you know, this was a huge opportunity. And so we designed this project to start with recruit kids with identified with copy number variants or genes that were associated with autism, cohorts of kids, all of them similar, characterize them behaviorally, take some skin cells, take these cells, make them into stem cells, take the stem cells, make them into neurons, and then characterize the neurons. Now, this was, from a practical point of view, a really tough thing to do because I was changing my field entirely. I mean, I had to give up, grants that I had on other things, and I had to, sort of come up with new things. And of course, you can imagine that the funding for this didn't go super duper well at first. I had no with good reason. My favorite one was when I got this review back, and one of the comments was, finding a defect in a neuron that is derived from a skin cell from somebody with autism, it's a hopeless task. It is like finding bin Laden. I feel so much better about that statement now, right? So we set out to do this, and as I'll talk about this tomorrow, but things have come a long way over the course of 4 years, and I should say this is only a little bit of it is me, right? I have very talented people in my lab, but I also collaborate with the Stanford Autism Center and people at NIMH who have helped us characterize and recruit kids, and with other people who are helping us to characterize the cells. So this has to be a group effort, and I think if we're going to do this in academia, this is how it has to be. It has to be a lot of people, right? So the goal is to identify defects associated with specific classes of autism. And as you'll see tomorrow, things have come along reasonably well, at least for some specific kinds of mutations. So for example, we have one population of kids that have a mutation in a protein called the calcium channel. And one of the things we discovered recently is that they produce too many neurons that produce noradrenaline. And adrenaline is this thing that puts you in sort of fight or flight response. It also makes you really anxious. And these kids are terrifically anxious. They have to be sedated to travel on airplanes. And so now we know why. Because they have about 4 times as many of these cells that are producing this thing that is making them nervous all the time. So the good thing is that now we actually have a target, right? And so we actually took some of those cells, and partly as a service to these families, and we screened them for all the compounds that have ever been used in man, right? Because we didn't want a new entity that had to be tested for safety. We wanted something that could be done now. And it turned out that we arrived at 2 things, one of which is now in clinical trials, right? So, there is a plausible direct path, at least in some sets of disorders. Another example, and I'll talk about this tomorrow, are kids with mutations in SHANK3 with Phelan-McDermid syndrome, and there we're not quite as far along, but now at least we know some of the deficits in the human cells. And we know actually that the deficits in the human cells are similar to some of the deficits in the mice, which means that the mice are actually good models. So we should work on the mice, because if we do something to the mice, it might actually work, because the humans look sort of like mice, at least at the cellular level. Yeah. So I think things are moving forward. As a parent, I always, this is sort of the point at which I want to always strike a sort of note of caution, right? I mean, scientists love to push their research as far as it will go, to make as many claims as they would like, and this is kind of in the DNA of being a scientist. But the deal is that when you're a parent and you hear that, and you hear somebody say, oh, we're going to cure it next year, it sounds terrible, because next year comes and it doesn't happen, right? And so I would say that we're making progress in the sense that now we have something to attack, right? We have something to grab onto. We have assays, and things are definitely moving forward. But the glass is, I'm not sure that it's even half full. It's a little bit full, and it's the wrong side of the glass. We want something to be closer to a treatment. But I think stem cells will actually provide an avenue to do this in sort of as part of, it's one tool in this sort of arsenal that includes, imaging and psychological testing and behavioral treatments and, and animal models. So, I see, at least I see some forward movement, and I see sort of a relatively bright future. So that's it from me. Thank you.
Okay, we're going to have one more scientist, researcher, come and join us, and this also brings me great privilege to be able to introduce Dr. Jim Perrin, who is, unlike the other two presenters who were PhDs, Jim is a medical doctor. He is a professor of pediatrics at the Harvard Medical School. He's also the director of the Division of General Pediatrics and the Center for Child and Adolescent Health Policy at Mass General Hospital, obviously in Boston. And he also is currently the head of the Clinical Coordinating Center for the Autism Speaks Autism Treatment Network. Jim has got a very long and successful history within the Academy of Pediatrics. He was very much involved in developing a lot of the guidelines for ADHD, which is another neurodevelopmental disorder. Jim kind of got dragged into autism about probably, 6 or 7 years ago. And for about 5 of those years, he kept saying, I don't know anything about autism. And I'm not allowing him to say that anymore. But Jim has been very much involved in the expansion of the Autism Treatment Network from the original 5 sites to now 17 sites. And Jim's going to talk a little bit about the papers and presentations that are, I believe, we didn't really talk about a whole lot, but talk about some of the great science that is happening in the area of treatment. So while Ricardo and his colleagues and David are actually looking for the underlying biology of autism, Jim and his colleagues are actually in a clinic taking care of children and adolescents and hopefully adults someday that have autism. So Jim, why don't you come up and join us as well?
I want to turn the page a little bit to a different kind of science that we're working on. But it's really so incredibly exciting to be at this meeting because so much is really happening. There are all sorts of great ideas out there that I think we can all put to use in our own lives to make the lives of people with autism substantially better in a variety of ways. I want to just start out briefly telling you about the Autism Treatment Network, which we began 5 or 6 years ago. We started with 5 willing centers around the United States who wanted to get together and develop some improvement in the care, some consistency in the care that families who are raising children with autism would get. And it was an effort to say, basically, we don't know what we're doing now, but we think we can do it a lot better than we're doing it now, and we think that families really merit comprehensive, multidisciplinary care and care that really addresses some of the issues that parents come to doctors about without being told, golly, gee whiz, it's just part of the autism, go home and we'll see you in a year. So a large goal of what we've done in the Autism Treatment Network is in fact to try to understand more about how to provide high quality comprehensive care, how to understand the fact that kids with autism and adults with autism have gastrointestinal disorders, have sleep problems, and these are all ones that we can really identify and we can treat. They're all things that can be worked on, made better. And that's in many ways the underlying goal of the Autism Treatment Network. As Peter said, we now have 17 centers around the United States and Canada. We're delighted with that growth. I'll talk more about that in a moment or two. And for a center to be part of the Autism Treatment Network, it has to agree that it's going to provide a model of comprehensive, ongoing, multidisciplinary, family-centered care. That's really our goal, that families will get a broad kind of care, which I think I could say honestly, the majority of families in North America raising children with autism are not getting that kind of care today. So that's really a key goal. And so you have to agree to do that. You have to agree to spend some time thinking together across the network about what are the problems we don't have answers to? How do we start addressing it? So it really began with some of our doctors and nurses saying to each other, I don't know how to manage this kind of problem when I see it in my clinic. And they're able to say to each other, well, this is how I do it. And there's been a tremendous amount of learning back and forth among the clinicians in the network about just how to deal with those kinds of issues. People have had to agree to enter at least 100 children per year into a common data registry that we have now. We actually have over 3,500 children and adolescents in the registry, which allows us to look at some of the things that are associated with sleep problems. For example, we have a lot of data that are in the papers being presented at this meeting that looks at the relationship of having sleep disorders with daytime behaviors, and the fact that not a great surprise to those of you who are parents or people with autism. You kind of knew this all along, that any of us who don't sleep well at night don't do very well the next day. Well, we've been able to document that. We've also been involved in research projects now to try to change that, to really develop mechanisms to address sleep problems in a systematic way for young people with autism so that they can sleep better, and by the way, their parents can sleep better as well. It's just one of the kinds of treatments that we're doing. We've also gotten all of our clinicians to sit down in groups and say, what are the right ways, given today's best evidence, to treat such and such a problem? So, should we be using EEGs in the evaluation of children with autism? It's a really interesting question. I bet you people in this room have a lot of different beliefs about what we should do about the use of EEGs, brainwave studies, in evaluating children with autism. We've developed clinical guidelines about the evaluation and treatment of constipation in children with autism. We're working on guidelines on the management of sleep problems in autism. So that's another agreement. If you're a part of the Autism Treatment Network, you got to be willing to spend the time and energy to develop the best possible evidence to support guidelines that we can use both in our own practices, but importantly that we can also distribute around North America, and hope that this will influence primary care doctors and subspecialist doctors working with families with autism as well. So those have been some of the underlying themes, this notion of a common evaluation of children, common treatments of children, a multidisciplinary strategy, a clinical data registry, the development of clinical guidelines, and importantly, about 3 years ago, and along with the wonderful support that we've had from Autism Speaks in developing the network, we were successful in getting a $12 million grant from the federal government, which allowed us to really develop a whole series of research projects within the network. And we've tried to elicit the views of families as to what are the kind of research that you'd like to have answers to. And that's been part of our strategy all along. We're really expanding that, in fact, in the next couple of years in a couple of different ways. And we now have 11 active research projects that are going on in the network that are funded through the federal grant. And we've involved over almost 1,000 children in the network in different research projects that are part of this activity. So the list that we just passed around includes, I realize not all the papers we have here. I realize we missed some of the papers that came out of the network here, but we have about 15 on this list. There are probably actually 20 or 25 papers that are network-related papers that are on the IMFAR schedule this year, which is really very excited about. There'll be 4 times as many next year, by the way. But it's really quite amazing. They vary, if you look through the list, from studies of sleep to studies of nutrition, critical questions that families would like some answers to. So we're getting some interesting data about nutrition. We're trying to understand more about what actually is the nutritional status of young people with autism in a very systematic way. We want to find out what the real deficiencies are in their intake and try to speculate what the implications for them and their families are of those deficiencies. We want to understand whether supplements are helpful or not, whether they really affect the deficiencies that we're defining in children with autism. And we're going to be carrying out some clinical trials of things like, for example, iron, because we found, and some of the papers you may have heard Ann Reynolds present, have to do with the fact that children with autism have higher rates of abnormalities in iron and probably iron metabolism. And that iron metabolism problems, by the way, are associated with higher rates of sleep disorders in young people with autism. So we're going to get involved, assuming we get our funding for this, we're going to get involved in some research which is actually going to be trials of whether adding iron supplementation to children who do have iron deficiencies really affects their iron, of course, but more importantly, does it affect their sleep too? So those are some of the projects that we're really working on. So it's really an exciting time. We're doing a lot of research that relates to I think, the issues that families would like to get some answers to. And I want to just talk for a minute about the future. So we are, through the 17 sites in North America, probably providing services to about 30,000 children with autism. So that's less than 5%, right? Okay. We have a long ways to go to getting to the point where we're dealing with providing high-quality, comprehensive, family-centered, coordinated services to the 750,000 children in the United States. And I don't actually have the numbers from Canada, but it's another substantial number in Canada too. So we want to figure out over the next few years, and we're really seeking to help with families in this, how to take this to scale. What does that really mean? How do we work much more actively with the primary care communities around our 17 sites, for example, to make sure that not only if you go to the center do you get high quality care, but actually in your community you get high quality care as well. So that's really a major goal of ours in the next couple of years' time, is to get this model of care available to substantially larger numbers of children in this country and their families. So that's what I had to say, and thank you all for your attention and support.
Okay, well, this is great because we got so much content. I'm going to actually, we were going to do just a little bit of Q&A with the scientists, but I want to make sure that our two stakeholders have an opportunity to share some of their perspective. And as we segue into that, someone over here made a mention or a suggestion to me, and that is, I think even though I'm not a scientist, I think I speak on behalf of the scientific community in that they are truly grateful that the families have entrusted the science community to do these types of experiments, to do clinical trials, to do research with their most precious thing, and that is their children. And none of this could happen if the families weren't willing to buy into that proposition of, we want your help, we need your help, we need to be part of the process. And I think that's kind of the spirit of which we come together today. And that is, there needs to be this sense of, we're going forward together, arm in arm, and we need to understand, and quite honestly, as we like to say, find the answers. Because those answers are going to help all of us, I think, live a better life. Obviously, our children or those that have autism, and also their related families. So I just think it's something that we all need to step back and appreciate, because I know the scientists and the researchers are truly grateful for the fact that we have that bond and that ability to move forward together to get those answers. So without any further ado, we're going to first have a parent, Sarah Logan, who comes to us from the Medical University of South Carolina. It's wrong on the thing. It's not the university, but the Medical University of South Carolina. She's a PhD student there, studying epidemiology with an emphasis in neurodevelopmental disorders. I'm sure some of that has to do with the fact that she's a mom of a 14-year-old son with autism. And Sarah's been coming to IMFAR for how long?
This is my second year.
Second year. So 2 years ago, she raised her hand and said she wanted to get involved in this committee. And so we're going to listen to a few perspectives from Sarah.
So as a researcher and a parent, I spend a significant portion of my life reading books and papers that have been authored by famous people, many of which that I've met over the last few days, so I'm a little bit starstruck at the moment. So I apologize for that. And I'm very also honored to have been asked to talk at this meeting and be a part of this committee, so I thank you guys for that. Just to tell you a little bit about myself, I am a mom, I have 4 children, and 14 years ago, my oldest was born. By the time he was 6 months old, we knew he was, I want to say, less than easy to be around, put it that way. And by the time he was 3, he was diagnosed with autism. And partly as I moved to prove that he was not autistic, I just started digesting and reading as much as I could and seeking out as many people as I could, just to know more. And as I was doing that, I was becoming fascinated with neuroscience and the human brain and ways to study neuroscience and autism. So to make a long story less long, I'm now a PhD student in epidemiology with a focus on neuroscience. But that's enough about me. I want to share a quote with you that Albert Einstein once said, that man should look for what is and not for what should be. And as a stakeholder, you know that it's natural and easy to become emotionally invested in things like this. But the striking thing about IMFAR is it's a place where people come together and they find that balance between emotionally invested as well as empirically driven to answer some very important questions. Whether you think autism is something that should be cured, whether misdiagnosed, if it should be embraced or celebrated or treated or grieved for, it's the one thing that we have in common is that we all want a better quality of life for everyone affected by autism, whether it's parents, family members, and those not so forgotten siblings. And the way that we can do that is to increase knowledge. So knowledge is power, and you guys are at an amazing place right now to not only gain knowledge, but share yours. I really encourage you guys to go out and talk to people. Just talk to people, and that's where it's going to go, and that's how we're going to get there. So that's really all I have to say right now.
Perfect. Beautifully said. Thank you so much, Sarah. And last but not least, we have another star amongst us, and he is an author. I'm sure some of you, if not many of you, most of you, have read at least one or two of his books. John Elder Robison is going to join us. He is the author of Look Me in the Eye and also more recently Be Different. John is an Aspergian, as I think as he likes to refer to himself, an autistic individual. And he's been a wonderful colleague of ours on the committee and has really brought some great perspective. And he's a lot of fun to be around. And he's a great speaker. So I'm going to turn the mic over to him. Are you going to come up here, John?
Well, one of the reasons that I think people invite me to participate in some of these legitimate scientific institutions like IMFAR here is that I bring a different point of view. And one thing I'd like all of you to consider at this moment is that as difficult as you may think it is to be an autistic person or to raise an autistic kid, right now, at this moment, there's 95 million pounds of water every single second passing Red River Landing on the Mississippi River. There is a 2,000-foot long thread of concrete called the Old River Control Structure, which is the only thing that is keeping the Mississippi River from changing course and leaving Baton Rouge and New Orleans as tidal backwaters. And you don't even hear about stuff like that in the news. It's like we hear about, killing this terrorist after 10 years. Nobody even thinks about that. It's like one of the biggest potential natural disasters the United States has ever faced. So anyway, when you think about the difficulty of autism, you think about how difficult it's going to be if New Orleans becomes a tidal backwater. And anyway, I know you didn't even care about that, but nonetheless, it's true. So I guess you wonder what my perspective is on this meeting. You heard from a few scientists about how we shouldn't be diverting our efforts to study children to study adults. And he made David made a well-reasoned argument that what we need to do really is study infants. We need to study earlier and earlier autism in children, not later and later in grown-ups. And while I think that that's a legitimate argument, certainly there's all the evidence we have says that the earlier we intervene, the greater the chance an autistic person will grow up to be different but not disabled, which is, I guess, how I am. And I think that's a great thing. But something I've realized the more I've gotten involved at looking at these therapies, these pieces of research that we're funding, there's a lot of talk that there's nothing being done for grown-ups. But the truth is, if you look at some of these good therapies that we're developing for children, if we look at this, there's a thing called Unstuck in there that's on display. There's a therapy called Peers on display. There are a number of therapies for teenagers. And every single one of those is applicable to grown-ups. So actually, I think that the idea that we're not doing enough for grown-ups, we're not applying what we're already doing. And I would urge all of you to keep in mind that many of the pieces of research that we do have applicability in areas that you don't think of. Another thing that I would like to urge all the scientists to think about is you look at this technology competition that we just had out there. And we had the first prize for the GoBug thing for tools to help remediate communication disabilities in people. And it was a kind of open-ended competition. You could interpret communication disorder any way you wanted, and you could come up with any kind of tool you wanted to do it. And the interesting thing is we got some valuable and worthwhile contributions from really industrial design students. Now, I would bet that there's not a single industrial design professional attending this IMFAR meeting today. And what that says to me is that there are a lot of other disciplines where we could be drawing expertise to solve these problems. We could be drawing in electrical engineers, computer scientists, industrial designers. You think, well, what's an industrial designer? Why does that matter? Industrial designers are the people who figure out where to put the buttons in automobiles so you'll press them, how to make a remote control that's easy and intuitive to figure out. Industrial designers are what made things like the iPhone what it is today. And so when we think then, how will we help people with disabilities? How will we help them not be disabled and function? One of the answers is industrial design. And I would certainly urge everyone to think about that. What I would like to say to all of you parents and stakeholders is that over the last couple of years of coming to IMFAR, I'm really pleased to see that we now have more and more. We have like neurodiversity bloggers, we have writers, we have people who would never have taken part in telling the story of IMFAR here covering what we're doing and talking about that for the rest of the world. I think that's really good. We also are drawing in autistic people. For example, I would draw your attention to Alex here. Now, Alex is a person with Asperger's who is actually doing all of the filming for this event. And I'm really proud of that, so I think you should all turn around, Alex, they can see you. So isn't that a great thing? We are actually drawing in our own community to help make this thing run. And the final thing that I would say to all of you who are, as you say, your parents, your people with autism, one great thing that you could do is go out and get involved. Don't just stand on the sidelines and complain about what's going wrong. Go out there and get involved with medical centers. Become a volunteer. Become a participant. Help support science. One of the criticisms that I hear as a science board member and as a person who advocates for science for groups like this, I hear all the time, well, we don't do enough community outreach. The thing is, community outreach won't work unless we have the therapeutic tools to help people. All the dollars in the world aren't going to make any difference teaching people with autism to talk unless scientists figure out how to teach them to talk. We have to have the science first before we can have the outreach. And there's so much criticism of science in the world, so many people saying, well, we need outreach. What we really need to do is we need to develop the tools to make outreach workable. And I would just urge all of you to keep that in mind, and I would urge all of you to do what you can to advocate for science and to sort of do your part and help out in this. Because when we have stakeholders, what we really need are invested stakeholders. And I would offer my own work as an example. I serve on science boards for all different organizations to help choose work that I think will benefit our autistic community and to help give a different perspective. You might not have appreciated the troubles with the Mississippi River, but there's more technically minded people that do, luckily. And so anyway, I would urge others of you to do the same. So just sort of go on out there and do what you can. And remember, if Old River Control washes in the Gulf of Mexico, you heard it from me first. So thank you all.